Starting with the first immunoglobulin, which is IgG. Remember that each IgG molecule consists of 2 L chains and 2 H chains, which are linked by disulfide bonds. And because it has 2 identical antigen binding sites, it is said to be divalent. The molecular weight of IgG amounts to a total of 150,000 Daltons. Now there are 4 subclasses of IgG, which are IgG1, IgG2, IgG3, and IgG4. These are distinguished by their differences in gamma chains. Now the most abundant of these is IgG1 and makes up 65% of the total immunoglobulins. Now with regard to the molecular weight, IgG3 is heavier than all the other IgG classes because it contains a long series of disulfide bonds in the hinge region. Coming now to some functions regarding IgG. IgG is the predominant antibody in the secondary response and constitutes an important defense against bacteria and viruses. IgG is the only antibody to cross the placenta, and only its Fc portion binds to the receptors in the surface of the placental cells. It is therefore the most abundant immunoglobulin in newborns. Newborns are actually not fully immunocompetent since immunoglobulin production begins at 6 months. So even though there are immunoglobulins present in the bloodstream due to the placental crossing of IgG, breast milk is a good source of antibodies in the first months of life. The IgG molecules from the breast milk are absorbed by the specialized epithelial cells in the intestine of the infant and passed on to the blood through a pH gradient. IgG is one of the 2 immunoglobulins that can activate complement, the other being IgM. Therefore, it is also involved in the immune complex in type 3 hypersensitivity reactions. Remember that IgG is also the immunoglobulin that is known for opsonization. Well, opsonization is actually a process in a host defense through which particles or complexes are made readily ingestible for the uptake by phagocytic cells. For a more detailed explanation, you can find the link to the video of opsonization in the description. Coming to the subclasses, IgG1 and IgG3 are more effective opsonizers than IgG2 and IgG4. IgG also has various sugars attached to the heavy chains, especially in the heavy chain CH2 domain. The medical importance of these sugars is that they determine whether IgG will have a pro-inflammatory or anti-inflammatory effect. Thus, IgG proteins specific for a single antigen that are made by a single plasma cell can possess different properties depending on these sugar modifications. Now, while discussing IgG, it's almost vital to mention protein A, or the virulence factor of Staph aureus. It is a part of the peptidoglycan cell wall and binds to the Fc portion of the IgG antibodies. This prevents opsonization, phagocytosis, and prevents complement activation. Now, IgG is also associated with warm hemolytic anemia, as it causes extravascular hemolysis, which is seen in conditions like systemic lupus erythematosus and chronic lymphocytic leukemia. Now, there's another condition related to IgG, which is known as common variable immunodeficiency, or CVID. It is a term that actually comprises a heterogeneous group of diseases, and most of which are idiopathic. But the patients with CVID present with low IgG levels and recurrent infections like sinusitis and pneumonia that are caused by pyogenic bacteria such as Staphylococcus pneumonia and Haemophilus influenza. However, the infections tend to be mild and may begin to occur late in life between the ages of 15 to 35 years, in contrast to other deficiencies, which we will actually discuss later in this lecture. The known causes of CVID are often functional defects in the helper T cells rather than the B cells. The number of B cells is usually normal, but their ability to synthesize the IgG and other immunoglobulins is greatly reduced. The intravenous gamma globulin given monthly reduces the number of infections.